Dear students, today we will discuss very important topic, that is BSc final exam topic, as well as today's lecture very important for all competitive exams. In today's lecture we will cover digestion of fats, lipids. प्यारे बच्चों, ये जितने भी हमने नोट्स बनाए हैं इन सब की पीडीएफ फाइल हमने वीडियो लेक्चर उसके डिस्क्रिप्शन में अपलोड कर दी है। सभी बच्चे अपने नोट्स डाउनलोड कर लें। Dear students, in today's lecture another final exam topic for BSc students, digestion of fats. Dear students, this process, this digestion of fats, lipids is carried by lipases enzymes, and this process, digestion of fats, that starts as well as completes in our small intestine. As in case of saliva, there is no lipase enzyme. It means in oral cavity, no digestion of fats, no role of saliva in fat digestion. In case of stomach, the gastric juice is present, and that gastric juice contains gastric lipase, but its action remains almost negligible, because in stomach, in gastric juice, the pH is unsuitable for the action of gastric lipase, as well as in stomach, no emulsifying agents. And for the proper functioning of lipase enzyme, suitable pH as well as emulsifying agents, it means this enzyme lipase acts upon emulsified fat. That's why in stomach, the role of gastric lipase that remains almost zero. Role of bile salts. In small intestine, three types of juice are present. From liver, bile juice. And that bile juice contains three important types of salts. Note down, NEET students, sodium bicarbonate, and sodium bicarbonate of this bile that involves to convert the acidic food, the chyme, into alkaline food. Chyle means from the stomach, the acidic food, that is chyme, and that acidic food is converted into alkaline food, into chyle. The alkaline food is chyle, and this conversion is carried by the bile salts, particularly sodium bicarbonate, that involves in the conversion of chyme into chyle, acidic food into alkaline food. And two another important bile salts, sodium taurocholate and sodium glycocholate. Dear students, in our bile juice, हमने कहा three important salts. Sodium bicarbonate that involves in the conversion of chyme into chyle, means acidic food into alkaline food, and two more important enzyme salts, and these salts are sodium glycocholate and sodium taurocholate. Note down, these two important bile salts are involved in emulsification of fat. These are emulsifying agents. Emulsification means conversion of complex large fat molecules into smaller units. This process is named emulsification, means conversion of complex larger units of fats into smaller units. And these smaller units are named emulsified fats, or these are triglycerides. And the enzyme, particularly the lipase enzyme, which acts upon the emulsified fat on triglycerides. Dear students, in our small intestine, three types of juice are present. The role of bile that involves in emulsification of fat, role of pancreatic juice, and that pancreatic juice contains main principal enzyme for the digestion of fat, that is pancreatic lipase. And this pancreatic lipase always acts on emulsified fat, means on triglycerides. And this pancreatic lipase, which is principal enzyme for the digestion of fats, it acts in 3 steps. Dear students, in 3 steps. First step, fat molecule in presence of bile salts, note down, sodium glycocholate and sodium taurocholate, and these bile salts convert this complex fat into smaller units. These smaller units are emulsified fat or triglycerides, and on these triglycerides, on this emulsified fat, this pancreatic lipase acts in 3 steps. Step number first, emulsified fat means this triglyceride in presence of this pancreatic lipase. Action number first, step number first, this triglyceride is converted into diglyceride plus 1 fatty acid. Step number second, this pancreatic lipase converts this diglyceride into monoglyceride and 1 more fatty acid. And step number third, this monoglyceride is further converted into glycerol and 1 more fatty acid. It means the emulsified fat, which is triglyceride, that is converted into 3 fatty acids and 1 glycerol molecule. And this process is carried by the principal enzyme. That principal enzyme is pancreatic lipase. And this pancreatic lipase always acts in 3 steps. Step number first, triglyceride is converted into diglyceride plus 1 fatty acid. Step number second, the diglyceride is further converted into monoglyceride and 1 more fatty acid. And step number third, last, this monoglyceride is further converted into glycerol and 1 more fatty acid. It means 3 fatty acids and 1 glycerol are end products of digestion of fats. Role of intestinal juice. As we know, in our small intestine, 3 types of juice. One is bile juice, and this bile juice having bile salts which play important role in emulsification of fat, mean conversion of complex fat into smaller units. And the principal enzyme, which is pancreatic lipase, that is present in pancreatic juice, and this pancreatic lipase always acts on emulsified fat, triglyceride, and converts this emulsified fat into 3 fatty acids and 1 glycerol molecule. And the role of small intestinal juice, that is saccus centricus, and in this saccus centricus, for the digestion of fats, intestinal lipase enzyme is present. And this intestinal lipase acts similarly to that of pancreatic lipase. Mean digestion of fat, very small amount of fat digestion occurs in our intestinal lumen cavity. It means the maximum, the main digestion of fat that takes place in the epithelial cells of, dear students, in the epithelial cells of small intestine. It means the digestion is intracellular. Very small amount of fat digestion in the cavity lumen of small intestine. Otherwise, maximum digestion that takes place in epithelial cells. It is intracellular. It occurs within the cells, epithelial cells of small intestine. Now, dear students, another today's important topic, that is digestion of nucleic acid. This is the last topic of digestion. Digestion of carbohydrates over, digestion of proteins over, digestion of fats we have discussed, and now the digestion of nucleic acids means digestion of DNA and RNA. And this digestion of nucleic acids also occurs in small intestine, because for the digestion of nucleic acids, saliva no role, gastric juice no role, as well as liver, the bile juice no role. Means in case of pancreatic juice and in intestinal juice, the enzymes for the digestion of nucleic acids are present. First of all, the role of pancreatic juice, and this pancreatic juice contains two enzymes. One is DNase, deoxyribonuclease, and the second enzyme, that is ribonuclease. These two enzymes, one acts upon DNA and second on RNA. DNA molecule, in presence of this DNase, deoxyribonuclease enzyme, and this DNA molecule is converted into deoxyribonucleotides, and ribonuclease enzyme RNase that converts this RNA molecule into ribonucleotides. And role of intestinal juice, and this intestinal juice also contains two enzymes, nucleotidases and, dear students, nucleosidases. It means the nucleotides of DNA and RNA, and these nucleotides, in presence of nucleotidase enzyme, converted into nucleosides and phosphoric acid, and this nucleoside is further converted into nitrogen base and pentose sugar, and it is carried by nucleosidases. It means the DNA RNA nucleotides are simplified by nucleotidases, and the nucleosides are further simplified by nucleosidases. This is all about digestion. Digestion of proteins, digestion of carbohydrates, dear students, digestion of fats, and digestion of nucleic acids. Thank you.